today we will focus on how Caltech scientists and engineers can work together to translate basic findings with platform technologies rooted in basic science, but to improve human health, and also with a focus on gap areas of opportunity. And this is important, and this is why we are starting the program today with a special 2025 David Dreier Lecture focused particularly on women's health. And I want to mention that although the Merkin Institute started in 2020 and we've been giving grants since then, in 2022 we were fortunate to receive a special gift from a longtime Caltech trustee and friend of Dr. Merkin to name a series of workshops and lectures. And today we are very pleased to host our third guest speaker in this series. And we also know that David Dreier is going to join us later today, so do say hi if you see him. And it is a particular honor to host our featured speaker for today, Dreier's lecture, Dr. Jennifer Garrison, that will be introduced by Dr. Mate Borsos. Mate?
Hi, good morning. This is my pleasure to introduce Jennifer Garrison, who is a pioneer, who's pioneering a global movement to advance science focused on understanding fundamental drivers of female aging. And she's doing this by co-founding and executively directing productivehealth.org, which is an ecosystem whose primary goal is to facilitate and accelerate translating scientific discoveries into products, diagnostics, and therapeutics to positively impact the lives of women around the world. So Jennifer leads the Center of Healthy Aging in Women at the Buck Institute, where she also is a faculty member with secondary appointments at the Department of Molecular Pharmacology at UCSF and at the School of Gerontology at USC. So Jennifer's research lab at the Buck studies how the changes in the chemical conversation between the ovaries and the brain may lead to the accelerated aging in women. Her goal is to discover and understand why ovaries age before any other tissue, and using this knowledge, extend the health span of women. Jennifer also recently redesigned and restarted the Aging Advanced Research Training course at the MBL in Woods Hole to train the next generation of scientists. And among several other awards, she's also a recipient of the Healthy Longevity Catalyst Award from the National Academy of Medicine and was named Allen Institute for Brain Science Next Generation Leader. So we're very happy that you're here, and thank you.
Hi, good morning. Thank you both for that kind introduction. My job this morning is I'm going to give a little bit of a schizophrenic talk because I want to tell you about what we built at the Buck Institute around women's health, but I want to also tell you how we got there. And it sounds like there's a real interest here in building something around healthy aging in women or women's health, and I'm really excited to share our experience. So for those of you who don't know, the Buck Institute is an independent academic research institute just north of San Francisco. So we're pretty small. If you haven't heard of us, there's no reason why you should have. We're only 24 labs. This is us. If you're ever in San Francisco or the Bay Area, you should definitely let me know and come visit. It's also an architectural destination. The buildings were designed by I.M. Pei. But the Buck really brings together a lot of different people. We have yeast geneticists working on cell biology, and we also have gerontologists who see patients. And the umbrella that holds us all together is that we're wholly focused on trying to understand mechanisms of aging. Now, I did not start working on aging. My Ph.D. is in chemistry and chemical biology. And then my training in my postdoc was in neuroscience. And so when I started my lab, I was really thinking hard about why would we study aging? And if you really stop to think about it, most of the chronic diseases of the modern world, the number one risk factor for everything listed here and more, your number one risk factor is your age. And so for heart disease, it's not your cholesterol or your familial history, it is your age. That's the number one risk factor for whether or not you're going to develop cardiovascular disease. And so the idea behind studying mechanisms of aging is that if we can understand the underlying causes, that we can exploit that knowledge to reduce overall disease risk. And if you step back and think about it, aging really is a public health pandemic at this point. We now have more people over the age of 65 on the planet than under the age of 5 for the first time since we've been recording it. So this is something that we have to start thinking about. And I should just say, in case you're wondering, we're not interested in extending lifespan. So this is not an effort to make everyone live to be 800. It's certainly a really fun and exciting philosophical discussion that I'm happy to have. But our goal is to really square off this curve of organ function over time. So what we want to do is essentially extend your health span, or the number of years that you're healthy. And from my lab, thinking about what I was interested in when I started my lab, I was really excited about bringing all of the work that I had done on thinking through neural circuits that control behavior. And my question was whether the area of the brain that I was excited about is the hypothalamus. This is really the control center for most of the circuits that control homeostasis. So things like energy and fluid homeostasis, temperature regulation, circadian rhythms, and also reproductive biology. And when I was starting my lab, there were a lot of labs that had just published really cool studies showing that with age, inflammation goes up everywhere with age, but in the brain, it's not uniform. So in the brain, there are hotspots of inflammation that happen during normal aging, and hypothalamus is one of them. And so, my thinking was all of the hallmarks of aging that we see with respect to, systemic physiology, they really do seem to stem from disruption in these homeostatic circuits. So my lab is really focused around trying to understand whether changes at the circuit level drive the physiology, and whether we can identify nodes in those circuits that change with age and target the circuit rather than the physiology to reverse or to slow down some of the things we see with systemic aging. And because of my background and the things that I was excited about, I'm really focused on bioactive peptides. So thinking through how changes in the long-range signaling between brain and the rest of the body change with age, and whether or not we can target those. So I won't go into this in detail, but I just wanted to put up here, peptides, bioactive peptides, are really essential for a lot of physiology, a lot of different behaviors, and when they're dysregulated, that can obviously lead to a host of different diseases. But I think about this long-range signaling between brain and the rest of the body as really how the nervous system encodes flexibility, right? We have the hard wiring, we have the activity, and this is a place where there's the ability to encode flexible modifications. And these are truly the most numerous signaling molecules in the CNS. And I would also say maybe something like the black matter or the dark matter of the brain, because we really don't understand how they work. So this is a summary of what my lab works on if you don't remember anything else. It's a silly cartoon, but I think it really encapsulates what we do, which is to think about how what I think of as the brain's Wi-Fi changes during aging, and whether we can intervene at different levels. So I won't go through this in detail, but for anyone who's interested in talking about this later, a lot of what my lab does is monitoring bioactive peptides in real time using peptidomics, and then pairing that with large-scale imaging in mice and worms, actually, to try to figure out whether we can do what I said we should be doing here. So I'm going to step back now and talk about something that happened about 6 years ago. We were chasing down the mechanism by which a particular neuropeptide was influencing aging, trying to figure out where it was working, and it brought us to the reproductive system. And I had not thought for 1 minute about my reproductive organs, beyond just the fact that I had a period every month and that I had a little bit of cramping or bloating or mood swings. I hadn't thought about it at all. And so I just want to bring you back to this graph. So what we're looking at here is organ function over time. And your organs don't age at exactly the same rate, right? But what you can say is that if you're lucky enough to live to older ages, most of the decline is going to happen in those last few decades of life. But there's really, when you look at reproductive organs, the female system really falls off this curve. So aging does happen in male reproductive organs. The difference is that it's synced up with aging in the rest of the body. For female reproductive organs, and I'm talking about ovaries, uterus, fallopian tubes, it really falls off the curve in terms of function. And this is important because as we're talking about extending health span and squaring off this curve, if we don't take this into account, then we're going to start to make inequality between the sexes worse and not better. So women do live longer than men on average. This is something that comes up a lot when I give talks. In every country where it's been measured, even in countries where the life expectancy isn't very high, women live longer than men. However, again, if they're lucky enough to live to old age, they will spend a significantly longer period of their lives in poor health. So in that period of time, we call it sick span or period of morbidity, that time towards the end of life when you have one or more age-related disease. So women are more likely to suffer from things like Alzheimer's disease, autoimmune dysfunction, osteoporosis. And this really is another way to say it is that at baseline, females have shorter health span than males. Let that sink in for a second. And it is directly because ovaries are aging faster than other tissues in the female body. We call them the canary in the coal mine for aging. So from a purely nerdy biology perspective, ovaries are actually a really cool system to think about because here you have a human model for accelerated aging, right? The longevity space has been looking forever for good models to use for testing interventions or therapies, and right now dogs are really the best model we have. But here's a model for accelerated aging in humans. But when a woman is in her late 20s, early 30s, ovaries are showing overt signs of aging, and the medical community actually calls them geriatric. So this has a really profound impact on health span. And I think it's important too to point out, as a woman, I hadn't thought about this at all, but ovaries are really important for more than baby making. So they're essential for fertility, no question about that. But they, like the brain, sit at the center of a really complex signaling network. And I don't want to trivialize it. I certainly don't want to oversimplify it. And we don't know much about these conversations, but the ovaries are signaling to and having dynamic conversations with every single one of the tissues listed here and more. And what we do know is what happens when they're not working properly or when you take them away. And so we can infer that what ovaries are doing, in addition to being essential for fertility, is that they're promoting health. And so 1 in 3 women have issues, either diseases or disorders, that are linked directly to poor ovarian function. We're all familiar with what happens with respect to poor ovarian function in a woman's 30s when the downstream consequences are things like infertility and miscarriage. But if ovaries aren't working properly at any age, that uncovers different health risks. And so for a girl who's 10, if her ovaries aren't working properly, that could uncover risk for issues going through puberty, or if she's gone through puberty, it could uncover risk for things like irregular menstrual cycles. For women in their 20s, poor ovarian function can uncover risk for things like polycystic ovarian syndrome, POI, primary ovarian insufficiency or failure. For a woman who's in her 40s, when ovarian function starts to fluctuate. So remember I said ovaries are sitting at the center of this complex signaling network. You're probably at least vaguely familiar with the symphony of hormones that we know about in the hypothalamic pituitary gonadal axis that are important for menstruation. Well, if you think about that, so if you just kind of click through the top level things that you know about, estrogen, progesterone, LH, FSH, maybe you don't remember exactly what's going where, when, but those are the canonical hormones that we know about that set up the cycle where, for the first part of the cycle, the endometrial lining gets thicker and eventually an egg is ovulated and then it's either maintained or it's shed. If you imagine the picture of those hormones and how they're fluctuating over time, that symphony, there's probably 200 or 300 other things that we know about now that are also fluctuating, that are signaling between the ovary and different organs. And so during perimenopause, when a woman is in her 40s and ovarian function starts to fluctuate, that essentially uncovers the risk for 135 and counting symptoms that we associate with perimenopause. And the reason that there's so many of them is because there's so many different things that can change in different directions. And this really drives home an important point about women's health, which is that female physiology is complex, and we need to treat it that way. And this is a place where individualized, personalized approaches are going to become really important. And then at and after menopause, which is really just a single day in a woman's life. Menopause is the day on which a woman has not had a period for 12 months. At and after menopause, when ovaries stop working the way they were before, it's important to say they don't stop working. They're still doing something. The reason we know that is because for women postmenopausal who retain their ovaries have better health outcomes than women who don't have their ovaries. So again, we don't know what's happening there, but we know they're still doing something. But at and after menopause, when ovaries stop working the way they were before, this uncovers risk for things like cardiovascular disease. That goes up fourfold after menopause overnight. Osteoporosis, autoimmune disorders, cognitive decline, the list goes on and on. And throughout a woman's lifespan, ovarian function we know is really, really tightly linked to metabolism and to immune function. Again, we don't understand these relationships, but all of this to say that ovaries are the architects of health in female bodies, and they are also setting the rate of aging. So we know that at least at a cellular level, if you buy into aging clocks, which is a different discussion we can have later, menopause essentially speeds up aging in a lot of different tissues. And so my plea to you is that after you've finished today, and maybe we've had some more discussion, that you think about women's health now through the lens of ovarian function, that this is really the thing that we need to understand in order to not just preserve health span, but to think about healthy aging across a woman's lifespan. I would also I'm begging you to stop calling them reproductive organs. I think that by calling them reproductive organs, we have essentially allowed the entire world to pigeonhole all of women's health through the lens of fertility. And of course they're important for making babies, but they are also doing all of these other things. And so call them ovaries and call them fallopian tubes and call it a uterus, but don't call them reproductive organs. So as a species, humans are really weird. We adopted this reproductive strategy that almost no other animals use. And in terms of a true menopause, there's humans, maybe some species of non-human primate, although that's controversial, and a couple of species of whale. Now you could argue that these are the best species of whale, right? Like belugas, narwhals, killer whales. But you can see that they're not even on the same branch of this particular phylogeny. And so it's evolved several times independently, but we don't know why we don't know why this is happening. And if we could understand the underlying mechanisms that are, causing these particular species to go through menopause, that would give us some clues about what's actually driving ovarian aging. There's also this really interesting correlation, and I stress it's a correlation. It's, this is on the statistical average, but life expectancy is actually correlated with age at natural menopause. So women who go through menopause later tend to live a little bit longer. And I would draw your attention to the x-axis here. So this is another feature of the complexity of female physiology, and that is that you'll notice that I've got a 14-year window here. And to be considered normal, age at menopause can span a 14-year window. So there's no other signature in all of human health that's so variable at the level of the individual. And again, if we could understand that inter-individual variability, that would tell us something important about the underlying mechanisms that are driving ovarian aging. For those of you who have a Y chromosome, I just wanted to point out that this is important for you too, not just because you have someone in your life who owns a pair of ovaries that you care about. Brothers of women who go through menopause later also tend to live longer, which means that there is at least some underlying genetic component here that's important for longevity in both males and females. So this is I love showing this movie. This is let's see if I can get it to work. Yeah. what we're looking at here is a cleared ovary, so the structure's up here in the blue. And what we're doing is driving through it so that you can see all of the features that are labeled with different fluorescent markers. And so what you'll see is the ovary is essentially a ball of balls. So in red are follicles, in pink are blood vessels, and you can see that the follicles span a pretty dramatic size range, and they're spread out as we're going through the ovary. They're spread out all over the place, and it's an incredibly complex organ. You don't really have to be an expert to appreciate that, but at the end of the day, this is really what we're talking about. And this cartoon really depicts the level at which we understand how the ovary works. And I will say that this particular cartoon is really just an average. And so at the level of the individual, this fluctuates and it's incredibly dynamic. But overall, female humans are going to have about 6 or 7 million eggs. Again, we have this weird reproductive strategy where we make all our eggs in one go, in utero actually, around 26 weeks gestation. By the time a female human is born, that number has dropped to about 1 million on average. And I would say this is the only part of the curve that we understand. And I can tell you about that later. It's kind of cool how this works. Eggs are the weirdest, most bizarre cells in your body. And this part of the curve really sets up how they're grown and how they go quiescent. Anyway, from here, by the time you might want to think about using your ovaries, that number's dropped to somewhere between 300,000 and 400,000. We don't understand that. And then this decline happens. So once you start cycling, about 1,000 eggs per month are lost. So 1,000 are recruited to grow, 999 die through atresia, 1 gets ovulated if you're going to ovulate. And then somewhere around late 20s, early 30s, this rate of decline sharpens until at some point the number falls below a threshold that isn't really well established. And then that coincides with age and natural menopause. Again, this is about how we understand it. So these are the questions that we think are really important to answer. What are the factor or underlying cues that are setting it in motion and driving it? Why is it so variable between individuals? And why does it happen so early? Why is it that menopause happens so long before end of life? The reason we are where we are is because there's been a dramatic lack of funding. So these are numbers from 2023. 2024 is actually worse. This is looking at the NIH budget. I expect this is going to get even worse. But about 10% of the overall budget was devoted to all of women's health from conception through death. And a large portion of this is really devoted to what I call the woman as incubator funds, which are very important, don't get me wrong, but I dug through Reporter to find all of the grants that I thought could fall into an ovarian aging category, and it's really just a rounding error. And this lack of investment is reflected in the biopharma pharma world as well. And on the private investment side, so looking at venture capital investment in all of women's health, it was actually going pretty well in 2023. It was much higher than it had been just 5 or 6 years earlier. But it still made up less than 4% of total investments in companies. And actually for 2024, this number's gone down to 2%, maybe because of the investment landscape. But this is ridiculous. We're talking about half the population of the world. And if you don't care about half the population of the world, if you're a business person, this is an amazing business case, which I'm happy to talk about more later. So this has led to a lot of consequences that some of you are probably familiar with. One of them being that a lot of the FDA-approved drugs that have been pulled from the market, generally speaking, if they're pulled from the market for safety concerns, it's because they had adverse effects in women, and that is because they were never tested in women. So the physiology of the female sex has really been understudied forever. The male body has been biology's baseline. I think everyone here who gets grants from the NIH is familiar with this metric that Only in 2016 were we mandated to include both males and females in our preclinical studies, but this is carried through into clinical studies as well. And I would just say you can't discover what you don't study. And so, from my perspective, we're not coming from, like, the data gap, it's not that we're a few years behind or that we're a few decades behind. We are generations behind where we need to be in terms of understanding female bodies. And so what we need right now are different models. We need exponential solutions. We need to bring as many people into the space as possible. Thinking about the drug pipeline, because that's something that's near and dear to my heart. This was a study that a review that a friend published, where she looked at the last 16 years of FDA drug approvals. There were 623 new drugs. She pulled out anything related to female-specific conditions and then got rid of anything that was a cancer drug. And we're left with 20 FDA-approved drugs in the last 16 years. Again, somehow we can't seem to break past this 4% number. But it's really shocking, and I think that we can do better. This is a look at the drug pipeline right now for female-specific conditions. This is a beautiful graph that McKinsey put together, and I love the way that they did this. So they've got the number of assets in the pipeline here. Oops, sorry about that. We've got the number of assets in the pipeline. And the x-axis is, on the left, things that kill you. And on the right, moving towards the right, this is how much suffering is this disorder causing relative. So mortality versus disability, essentially. And you can see that we're really good at making cancer drugs. But the things that are related to what I was just talking about with respect to ovarian aging, these things are out here with very few assets in the pipeline and maximal suffering. So this is really what's been driving me for the last many years, is that not just is the pipeline empty, but it's, like I said, we're coming from generations behind. There is a great business case for this, though. So for anybody who wants to talk about this later, I'm happy to talk about it. 6 years ago, we decided, we got a seed grant from the Bia Echo Foundation, and we decided to start the Center for Healthy Aging in Women at the Buck Institute. And our goal here is to do this research, basically to fund innovative solutions to try to understand what's driving ovarian aging. And I think given the multifactorial nature of the way that ovaries function, we really thought that we should be bringing in as many people from different areas of science as possible. So it's made up of faculty who are at the Buck, some adjunct faculty who are at other institutions who we think have really unique perspectives that strengthen what we're doing. We have an evolutionary geneticist who's actually studying that basis for the difference between menopause and not menopause in whales, which is really cool. And we are about to open a search for a new junior faculty member, if you know anybody who's on the job market. But our goal is to fund things that are maybe not being covered by governmental funding or that are too high risk or out of scope for that. And the kinds of projects, you can read about this on our website. I won't go into this in too much detail, but I put this up here because I wanted to try to frame just how little we know about ovarian function. So I'll tell you in a second about some of the science that's come out of the center. And one of the great movements forward has been applying multi-scale omics to ovaries. And one of the challenges there is that ovaries are really dynamic, right? So they're changing not just on the time scale of a cycle, 26 to 30 days, but they're changing on a time scale that's much bigger than that too. So if you think about the development of a follicle, from a primordial follicle that's sitting there and that's quiescent all the way through a follicle that gets ovulated, that time scale actually from recruitment to ovulation in a human is over a year, okay? So every month, 1,000 primordial follicles are recruited to grow. And so, and it takes over a year, and so there's those things happening. And again, there's a lot of death through atresia at different points along that curve. And then you have the time scale of a cycle, right, which is 26 to 30 days, and that's kind of the tail end of this process. But at any given time, you have all these different structures inside the ovary, and they're dynamically changing. So there's like a macroscopic physical remodeling that's happening every 30 days. That's crazy when you think about it. But beyond that, the cells that make up the follicle, so everything except the egg, they transform, they change not just their shape and they proliferate, but they change their cell state and their identity. So they can go from something that looks completely different from an omics perspective, but it's the same cell. And so applying a lot of these omics technologies has been really challenging. And in fact, we funded the first single-cell sequencing data in ovaries 5 years ago. And last fall, we had to have a cell types jamboree where we brought together 60 scientists from all over the world who've been collecting omics data on female reproductive organs. So that's fallopian tubes, uterus, and ovaries. And the reason we had to have this meeting, and we were lucky that CZI funded it, is because we literally sat together in a room for 4 days to agree on what to call a cell type in the ovary. The cell ontology has not been set. And so that means that you can't compare across these large data sets. That's where we are. We also don't have great models. Thinking about female bodies as a whole system rather than siloing things into organ-specific or disease-specific buckets. And then thinking through, like, what are those signaling molecules that ovaries are making? This is what we're doing. Almost immediately, when we started the center, it became clear that we could convert the entire Buck Institute. We could get all of Caltech, all of UCSF, all of Stanford to focus on this problem, and it wouldn't be enough. We need an army, like literally an army of creative scientists to work on these questions if we want to slow down or even reverse ovarian aging. And so we started almost immediately a global consortium. And the global consortium is really meant to stimulate research outside of the Buck Institute. And I won't go through all of the things that we do, but our goal is to try to, as we're building this new field that's at the interface of reproductive biology, which is a mature field that grew out of developmental biology, and longevity science, that as we build this new field, that we think hard about how to not just have the discoveries, but also how to translate them as fast as possible. And so that means forcing conversations between groups that don't normally talk to each other. So we spend a lot of time getting clinicians to talk to the basic scientists, bringing in the funders, the early stage founders for the biotech companies. We don't have any IP that would go towards a true intervention or something that would be like a real drug yet. But what we do have are what I call Band-Aid solutions. And those Band-Aid solutions that are in development are really important. So we spend a lot of time building infrastructure, thinking about advocating for change, although we'll probably pause on that for a bit now, and creating a community that really supports translation in a way that typical academic science doesn't. So we funded grants. We've done 2 rounds of grants so far to 49 scientists all over the world. We support different career stages. We encourage people to take risks. We have absolutely no idea where along that trajectory the causal things that we care about, the drivers, are actually happening. And so we fund research really broadly. And we spend a lot of time thinking about how to do the review in a way that maximizes everyone's time and biases against institutional and gender bias. So this is what our network looks like right now. We do have, so Mate, we consider him part of our network for sure, because not only does he attend our reproductive aging conference, he was a speaker last year, but he's also a collaborator. So hopefully he'll talk a little bit about that work later. These are some of the studies that have come out of just the center at the Buck. And this is what's happened to the field of ovarian aging, at least from the perspective of citations. So directly from our grants, we've had over 50 papers published since 2020, which is when we first started this. And this is just looking globally at the citations. And I made this slide in July, so I fully expect that this number's up here now. And I think I'm really proud of this, and I think that there's a blueprint here for how to do this kind of work in a way that really pushes forward as much innovation as possible. This is just a few examples of some of our grantees, what they've used the money for. So Holly Ingram is an amazing scientist at UCSF who's been working on female reproductive hormones for her whole career. And she used our funding to find a novel hormone that builds bone. And this is actually being translated now into a company, and so this is a real target that's filling the pipeline. We also funded Yushin Sa at Columbia and one of her postdocs, and this has been parlayed into the very first clinical trial that's ever been conducted. And it really doesn't matter what the outcome is here. What matters is that this is a paradigm shift in the way that we approach clinical studies in females. So they're looking at ovarian endpoints not for fertility, but for aging. And then I'll tell you very quickly about a collaborative project that Allison, who's a postdoc in my lab, took on with Hattie Chung, who at the time was a postdoc at the Broad and who now has her own lab at Yale. It's a huge collaboration with Fei Chen, Pardis Sabeti, and Alex Schleck's lab. And really what we were doing is just asking whether we could get some spatial resolution across the ovary. So we use SlideSeq to look at mouse ovaries across young, middle, and older animals. So this is old reproductively speaking. And we profiled estrus and metaestrus because we wanted to look at the period of time when the most remodeling is happening. And we spent a lot of time thinking through how to capture as many features in an ovary as possible, keeping in mind the spatial, not important for this, but keeping in mind the spatial relationships between follicles and things like that. And what's beautiful about this is that you can see without any real analysis, you can see that the data really reconstructs the follicles and the corporal lutea, which I won't talk about because it's too repro-specific right now, but we were able to find a lot of different cell types that map back onto the structure in a way that's beautiful. It really illustrates the power of this technique. And we were able to then assign cell types to follicles. And just having this data really gives us a picture and it's generated so many hypotheses about what's happening during this remodeling process that I can't even tell you. And I'm going to go through this quickly, but this paper is online, so you can find it on bioRxiv. But overall, we were able to collect 653 follicles and 234 corporal lutea. So from the left here, each one of these is a puck. So these are all of the reconstructed spatial data. And going in this direction, this is from looking at folliculogenesis. So this is from primordial follicles all the way through ovulating follicles, all the way through early and late corporal lutea. And I think it's just a beautiful piece of art. The other really cool thing that we were able to do was to collect 300 oocytes. So, with oocytes, typically in single cell data, you get rid of them because they're too big. And if you do happen to catch them, you don't know where they came from. And as you can see, the heterogeneity of what's happening in terms of what follicles look like, right? Every single one of these that's in green presumably contains some sort of egg. And so what we were able to do was to not just profile the oocytes, but then map them back to the follicle they came from. So it's just, I can't even tell you how amazing it is to look at this data every day. These are some of the conclusions that we came to from our data, and I'm not going to walk through them because I know I'm running a little bit long here, but you can read about this online. I wanted to take a minute just to tell you about some of the resources that we built, because hopefully people in this audience will want to use them. So one thing we thought hard about was how do we get the most creative scientists who are already established in other fields to start thinking about ovarian aging or just ovarian function in general in their own work. And so we started a reproductive biology core facility. So literally an ovarian biology core facility, like a proteomics core or a microscopy core, that is staffed by an incredible Ph.D. scientist who was actually one of our postdoctoral scholars from Diana Laird's lab. She is directing this core in a way that, yes, they can do fee-for-service experiments in rodent, non-human primate, and human ovaries, and other reproductive tissues as well. But she's also got a team that can partner with you, that can collaborate with you, that can, if you've never done experiments in ovaries before, help you think through not just how to collect the data and what to do with it, but then how to think about the follow-up experiments. And we're running, probably this summer, we're gonna run our first boot camps, which are structured so that you can send your trainees to us for a week where they get intensive training in the techniques, and then they can bring that expertise back to your labs. And if you happen to have models where you think the ovaries are interesting, we have this campaign called Save the Ovaries. We also have a tissue database of reproductive tissues that we maintain, and all you have to do is use one of our standardized protocols to preserve the tissue and store it. And if someone wants it, then they pay for the shipping. And so we maintain the database, and if you have any tissues you want to contribute, please let me know. We also started the first international conference on this topic, and we've had 3 meetings that were all supported by NIH grants. And what's amazing about this, especially the first one, was that I didn't know if anybody would show up because there wasn't really a field at the time. We started in the middle of COVID, and it was unclear. Most of these people are not our grantees. It was unclear whether people were really gonna want to come together. But what's amazing about each one of these pictures is that there's scientists here, but there's also a lot of clinicians. There are VCs in this picture. There are biotech founders. And so this Repro Aging Conference has really become a hub for that kind of forcing communication that I was talking about earlier. The next one is going to be in May, May 4th through 7th in San Jose. So if you want to come, we would love to have you. But more than that, this is meant to be a place where we brainstorm ideas. So at the end of every one of these conferences, we've had a brainstorm session where I ask everyone who's there, like, you're the field. What resources do you need to move things forward? And a lot of the things we've built are based on those conversations. So this is one thing that we have under development from one of those brainstorm sessions called a Knowledge Hub, which is meant to be a place for women, for scientists, for clinicians to come for information that'll be built on multiple layers, so Wikipedia style at the top, where we're not going to make all the content ourselves, but if we source out, the promise is that it's been vetted by a scientist or a doctor. But then clicking down one level, because this is such a new field and that we hope there's going to be an explosion of work in the space, we spend a lot of time at that middle level putting together a way to characterize the papers that are being published simply, where it's, here's what the study was trying to do, here's what it actually achieved, here are the questions that it leaves open or that it answers, and then for non-scientists, non-physicians, if there's anything actionable from that study, here's what it is. But it's meant to be a trusted source of knowledge, something that right now, double-edged sword, because we have so much work happening in the space on the private side, that right now if you go and search for PCOS on Google, for example, what you'll get is 50 factoids that if you click on any one of them will take you to a page that's trying to sell you something, and I think that's a real issue. The other thing I did recently, which was really fun, I'm on the, everyone here is probably familiar with the X Prize because of where you're located. I'm on the Health Brain Trust for the X Prize, and so we get together in March and we sort of bat around ideas for what the next X Prize should be. And this year I ended up competing with an idea for the X Prize for Women's Health, and we actually won. And so among all the ideas from all the different domains, the double X Prize for Women's Health won and will be developed into the next X Prize. So it will be the biggest X Prize that's ever been done. So they have a longevity prize running right now that's 100 million. This is going to be at least 150 million. And I love this because the X Prize will bring in interest and people and companies that wouldn't otherwise think about women's health. So I think it's another part of the ecosystem that needs to be developed, and I'm glad they're going to do it. So I'll just end by saying, what we fund dictates what we know, and what we need right now in women's health is more discovery-based research. And this is not about delaying menopause, right? I'm not talking about getting women to have babies when they're 70 or extending periods to the end of a woman's life. What I'm talking about is preserving ovarian function, the endocrine function of ovaries, preserving that health-promoting part of ovarian function. And I'm not talking about femtech either. So I don't think I need to tell you why this is important. I will point out for you guys that we really need ambassadors. So it'd be great if you told people about what we're doing, and get in touch if you're interested in learning more. I have to thank, we have a wonderful scientific advisory council that chooses the grants, so that's not me. Alison Rodriguez does almost everything for both my lab and for the consortium in the center. Frida Poli was actually my entrepreneur in residence, and she recently got poached by MIT to start a center for behavioral neuroscience and AI because of her background, but we're actually secretly, not so secretly, using it to do women's health and AI. So if anybody's interested in that, let me know. And then our donors, everything we do is through philanthropy. And these are the acknowledgements for my lab, my funding sources, my collaborators. I didn't talk about a lot of the work in my lab, but again, Allison, and here's my contact info if you would like to get in touch. So thank you. And I've purposely left a lot of time here so that we can have a discussion, hopefully, if you have questions. There's lots of things I could go into more detail on.
We have microphones that,
I can also repeat the question if there's people online.
I have a microphone. I don't know if that's reasonable. Down back here. Greetings. There's a little bit of a delay here. So I'm glad you made that comment at the end, because I think it may not be clear to someone as young as you, but there's actually quite a lot of good life left after menopause. So the question is, do you guys have the bandwidth to also look at things like bone density and heart health and also neurological disease? Because I think, and also autoimmunity, which is a tremendous problem for women over men. I don't know what areas you're prioritizing at the Buck besides the ovarian stuff.
Well, actually, everything you just mentioned, I don't know if you were here for some of the early slides, but everything you just mentioned, all of those things are linked to ovarian function, right? So the question is so the questions are very, very clear, like, why is it that women are more susceptible to things like Alzheimer's disease or autoimmune disorders, and that both of those things are accelerated, or the risk goes up at and after menopause. And so, we fund research across the board into all of those areas. At the Buck itself, my lab, I'm a neuroscientist, so I'm really hyper-focused on thinking through what are the signaling molecules that are changing, what are the ovaries producing, step number one, and then how do they change with age? And that by itself is really fascinating, because at least the kind of signaling that we care about really mediates the crosstalk between the brain and the immune system, but also between the ovaries and the immune system. And a lot of the data that's come out of the single cell work, not just ours, but a lot of other people's work, and also some of our grantees, I should say, has really, we've gone a long way in the last 5 years learning about what seem like simple things, but they weren't known before. Like, for example, that ovaries, with age, the biomechanical properties of ovaries change. So they become stiffer, which is due to an increase in fibrosis and a decrease in hyaluronan. And it does seem like, based on some of our grantee work, that antifibrotic agents can reverse that. Now, obviously, antifibrotic agents as they exist today, that's not going to be a solution, but it does give you a whole host of characters to think about in terms of potential interventions. And then on the low-hanging fruit side, we funded a follow-up grant to ask whether we could adapt shear wave elastography, which is a type of ultrasound that is used in other tissues to measure biomechanical properties, whether we could adapt that to follow ovarian stiffness in humans and use that as potentially a biomarker for, it's not going to cure anything, but if you had a biomarker that it would at least tell you where you are on that trajectory, that would be really profound. We have a lot of different things that we're working on.
Yeah, I just wanted to mention that in immunology, there's a tremendous amount of interest in sex-linked diseases because many immunological genes are on the X chromosome and they're not completely dosage compensated. I'm not sure that you're completely connected with that group, and you might really want that liaison.
Absolutely. Actually, one of our grantees, Berenice Benayoun, who's at USC, this is what she works on, is sex-specific differences in the immune system. But yes, I would love to meet more. Immunology scares me, to be honest. But thank you for the question. And I'm sorry I didn't spend as much time. I took a lot of my slides out that were about menopause. But it is really important to say, number one, that we're not trying to end or delay menopause. That's like a nonsensical statement. Unfortunately, the popular press does tend to pick that up as the take-home message. But also that ovaries don't stop working. It's not like they stop working. They just stop doing what they were doing before. They're still doing something. And that is an area of active research that could be really fruitful, not just for thinking about ways to mitigate those impacts on postmenopausal women, but again, getting to this really basic question, which is how, it's the most basic question, how does female physiology work at every age?
Can you hear me?
Here.
That was a stunning talk. I am really happy to also see this conversation about women's health amplified. One of the reports that I read recently is that the age of menopause onset on average has been postponed, generally just because of modern medicine or so on. But it's also the age of menarche, which is the onset of menstruation, has been delayed. And that makes me wonder, in the data that we see, women who have late menopause and live longer, do they also have a late start into menstruation? Is it the reproductive timeline is the same but just shifted?
Yeah, no, that's a really good question. And the answer to a lot of these questions is, well, we don't really know. But for some questions, we do have data sets. So age at onset of menarche and age at natural menopause don't seem to be correlated. So there's this, there was this fear that as we're seeing girls go through puberty earlier and earlier, that might mean that the age of menopause would move back and back, but that doesn't seem to be the case. In fact, the age of natural menopause hasn't changed very much since we've been measuring it. Now, that's with a caveat that we don't measure very much, right? I mean, part of the AI studies that we're doing now are horribly complex because we're going into all the EMRs that exist in the US, so millions and millions of amazing rich data sets where nobody has actually documented age at menopause. There's very few data sets where that's documented, and so the question is, can we use all these new technologies to go in and find that information either through unstructured data, or can we infer it based on other things? And so age at menopause isn't something that's been really well documented. But the other thing to say is what you said is a really big misconception. So it's not that suddenly the Industrial Revolution made it so that people live longer and therefore our reproductive span just hasn't caught up. That's not accurate. We have an evolutionary geneticist at our center actually who wrote a book about the evolution of the female form, which is fascinating and really meant for lay audiences. But this is not my area of expertise, so I'm not going to dive into it too deeply. But essentially, what she'll tell you is that humans have been living to 70, 80 years old for millennia. So 70,000 years ago, maximum human lifespan was approximately what it is today. We might have gained 5 or 10 years, but really it hasn't moved. What changed was that the Industrial Revolution and modern medicine have essentially made it so that infant and childhood mortality have mostly been abolished. And so it's the average age that's changed, not the absolute age. And so it's not so simple as just we're living longer and our reproductive span hasn't caught up. And the underlying evolutionary reasons why age at menopause happens when it does in humans that's an open research question. And there's lots of different theories for why it might be, but it's not something simple and straightforward like the grandmother hypothesis. That might be part of it. But if you talk to Dina, her name is Dina Amira, what she'll tell you is she thinks it has more to do with mother-infant conflict, maternal-fetal conflict, which comes back to some things that I'm not going to talk about here, because again, it's not my area of expertise. But it's not so simple as that.
I do wonder that, because modern medicine, quote unquote, has not been geared towards improving, health of ovaries, for example, in the last millennia. So ovaries have not been able to catch up to the same way, that the other body has. The discovery of vitamins, improving health, etc. We don't have that same kind of thing for ovaries.
Not yet.
So there's definitely an evolutionary constraint, but I wonder if it can still be improved. Thank you.
Hi. So there was a big change in medical practice at the turn of the century with the rather sudden diminution in use of hormone replacement therapy. And the question, I mean, there are many questions about that. The impact on AD is one of the open questions, and presumably you're kind of looking at that. But do you have access to enough samples that actually do experience HRT? Because I'd be very curious about what the ovaries are doing under the influence of HRT for different lengths of time out. So are you able to look at that, or?
So yes, and also. So what you're referring to is the study that was published in 2002 using the Women's Health Initiative data set, or the WHI study is what people call it, but I think it's important to point out that the WHI study is a longitudinal, multi-decade study that is an incredible data set, and there's a piece of it that continues even today, and literally thousands of papers have been published using those data. And so unfortunately, the first study that was published in 2002 was deeply flawed, and there have been books written about this, and lots. You can go read about it if you're interested, but the take-home message is that everything that it said was wrong. And none of the data, it was structured wrong. The study was there were so many issues with it, but also what they reported wasn't even statistically significant. So,
I know.
I mean, it's crazy.
I wanted to let you kill it.
It's crazy. So I'll just say it here. I'm not a medical doctor. I don't give medical advice. I'm a Ph.D. scientist, but I can tell you what the data says. And hormone therapy does not cause cancer, breast cancer. It does not. And so hormone therapy is by far the best Band-Aid, and it is a Band-Aid that we have to mitigate some of the issues that menopausal women are facing. And until we have something better, it's by far the best thing we have at all. The other thing to say about it, though, is because it's something related to women's health, there has been almost no innovation in hormone therapy since it was introduced in the 1940s. And so, the one piece of innovation has been changing from oral estradiol, which can, when it's metabolized by the liver, can induce clotting factors, to adapt it to a patch, which bypasses metabolism in the liver and is safer. So that's great. But right now, hormone therapy is typically estrogen, progesterone if you have a uterus, and maybe testosterone, but mostly not. And the number of, it's no longer under patent, obviously, and so the number of possibilities for titrating the relative amounts, the total amounts, personalizing it, it's really not there. And so it's administered kind of like a sledgehammer. And that means that a lot of women who start it experience side effects and then they go off of it. And that's tragic, because having estrogen around, we know, maintaining estrogen levels is super important for bone health. And so if you don't do hormone therapy, you absolutely will get osteopenia and osteoporosis if you live past your 60s. And then, talking about things like cardiovascular disease and cognitive decline and all these other autoimmune disorders, those are links that we're still mining the data for and collecting the data to really establish in a way that's profound, but you can make those inferences. And so, sorry, I lost your question.
And for AD, it's actually a pretty strong inference that there is impact if started early enough.
Yeah, but it's, again, it's a Band-Aid. It's an imperfect Band-Aid, but imagine that we understood enough that we could, layer on another 5 components.
Yeah, I'm sold. That's why.
And that's not even, that's just the low-hanging fruit. There's so much opportunity here, not just for businesses and thinking through innovation, but just to improve the lives of half the population.
Sorry. So I would be excited to know if looking at people now, even the relatively few that are getting HRT.
Oh yes, sorry, coming back to your question. Gotcha.
How does that cycle back and impact the ovaries that are there? Because they normally wouldn't be seeing elevated estrogen, progesterone.
So yes, we're working on, actually a few different labs that we funded and some other labs that aren't in our funding stable, and also our collaboration with MIT. We're trying to get enough data to really solidly make those connections, because the problem is, because hormone therapy it's applied in this kind of haphazard way, often not documented. Yep. It takes a long time for someone who starts hormone therapy, typically ends up changing things a lot before they find a combination that works for them. And so it's really hard to find enough people who have similar treatment regimens at this stage. The bonus, or the thing that's happening right now, is that there's so many women who are demanding better options. And there are a lot of startups and also concierge medical clinics that are starting to not just give women hormone therapy, but also to collect really careful data, because they want to contribute to scientific knowledge and innovation. That those data sets are coming online, but they're definitely not there yet.
Cool. There's hope. Thank you.
There's hope. Oh yeah, it's a positive message. Don't get me wrong.
Hi, Dr. Garrison. Oh, okay, it's on. Thank you so much for doing what you do, also for being here and for the talk and discussion. I guess the most big picture way I can ask my question is, just referring to your slide where you show the different rates of aging of different organs, and thinking about the ovary and how dynamic it is over the life cycle of a human female. Do you think that it's more a question of rate, question of things speeding up and slowing down, or do you think that it's more of a question about mechanism and the mechanism of aging, or the definition of aging changing over time? And I guess the follow-up question to that would be, how, from a position like yours, do you ensure that it's really like a patient-centered, directed research initiative or investment to try and solve the problems in order of what's most important to patients and how they experience their lives?
Yeah, okay. Well, that's 2 very different questions. Let me start with the first one. Sorry, remind me again.
The mechanism versus the rate.
Yeah, that was really insightful. You've obviously thought a little bit about aging before. And I will say I'm, how can I say this without insulting all my colleagues? I'm a skeptic. And so I think it's probably, I think what you said is spot on. It may very well be about the rate of aging. For the very few pathways that we know about that impact aging in the rest of the body, they are also certainly operating in ovaries, right? Things like mTOR signaling and NAD signaling. And when you modulate them, it can have a beneficial effect on ovarian function. How you design, this is something we thought about a lot, how do you design a clinical trial in a woman who's in her 30s that only runs a couple of years, but then gives you some output that's going to tell you something about either the rate of aging or really the endpoint of ovarian aging? But I also think that there are probably ovarian-specific mechanisms that are operating there. And again, it doesn't come down to something really simple. I wish it had, like damage, because if you imagine over a female lifetime, assuming that ovulations are allowed to occur every month, somewhere between 300 and 400, right? But if you look at ovaries, like if you look at cross sections of ovaries, and if you look at ovarian tissue, right, when ovulation happens, it's really dramatic, and the amount of damage that has to be repaired is pretty profound. And obviously the ovaries have really evolved to take care of that pretty efficiently, although, part of the reason that we see probably a huge change in immune cell infiltration and inflammaging, we call it, but inflammation that happens during aging in the ovaries might be related to that damage. But that's not the thing, right? When we change fibrosis, when we get rid of multinucleated giant cells, when we do all these other things to modulate immune function, it has some impact, but it's not the whole thing. And so I think it's probably going to be a mixture of those things. And then in terms of prioritizing research focus areas to think about what's going to be the most beneficial for patients, we're coming from a place where we know nothing. And so at this stage, we're funding research that's really across the board. It's, the only the boundaries we've put so far on our funding is we don't fund clinical trials directly because, we don't have that level of funding. And we also don't fund things that are specifically and directly about improving IVF. Or really about fertility outcomes. Because while that's one way to measure ovarian function, that's really not what we're concerned with. And I feel like most of the research dollars that have existed, paltry as they are, in the past have really gone towards things like that. So those are the two bounds that we put on it. In terms, though, of thinking through how to maximize what's coming out of those basic science questions and experiments, we do have a lot of resources for our grantees and also for the field. We've got a translational advisory board that helps us look at the science that people are doing, and if something's translatable, to help them sort of think through what that might look like, even if they're not interested in translating it. And then we have resources to put towards getting it translated. We also have spent a lot of time thinking about how to fix what's really a problem in biotech development in general. So it's like there's two punches that happen that really, I think, lead to a lot of companies, and we're watching this now with companies that started 4 years ago. Perfect science, amazing business model, like doing all the right things, and they still fail. And that's because on the academic side, we have to publish sooner than you might want to in a perfect world, because we have trainees and junior faculty who need for their careers to publish papers. And so that means then that you have to file the IP maybe earlier than you would otherwise. So it comes out of a protected space too early, and then that essentially drives people to get VC funding earlier than maybe they should. And all of this adds up to too short of a runway for the development of something that is essentially being funded by a model that was for tech and not biotech. Anyway, we've thought about this a lot. So we have a bunch of resources that we try to facilitate translation in a way that hopefully mitigates some of those issues. Yeah.
Hi, George Salmazenko, Executive Director of Merkin Institute. On behalf of all of us, I want to thank you, Professor Garrison, for a wonderful talk and excellent questions. We have a few minutes before Professor McMahon comes up and gives our next talk. You want to trade order? Okay. You may applaud now, right?